That's okay. Yeah. Let me get this to you. All right. So we are going to start our unknown labs on Wednesday. We will have lab 13, which is ELISA, somewhere like in 2 weeks or so. So we will do that. But just wanted to introduce the unknown labs to you guys. It is not a difficult lab. It's actually really fun. Things work out pretty good. Every person is going to get their own bacterial culture in a form of a slant. So each person is responsible for their own unknown. Having said that, does that mean that you can't discuss with others, get their opinions, talk to me about it and all of that? Not at all. You can just talk, look at your notebooks, all of that. We can have a discussion. But I'm hoping with what I go through today, that it should be easier for you guys to get it started. This is chapter 15 from your book, which we don't cover. The chapter 15 was diagnosis. And why I use it is because basically what you're doing with your unknown is almost the same as what we do for a patient that comes in with an infection and you want to diagnose and figure out what is the infection that is causing them the problem. When you do that, the patient is basically, you're going to get a sample from the patient, and the samples are different. You know, it can be blood, it can be skin, it can be sputum, it can be urine, fecal matter. So all of these variety of different samples depending on where they have the issue going on. Biopsies, again, so you can have all these varieties. But what you get to do is when you get it and it's an infectious disease, then it goes to the laboratory and they have to grow it in a lab, okay? Obviously for some of these samples that you're taking, proper techniques needs to be used, like a blood will coagulate immediately. They use a special tubes, special chemicals in order to get the blood so blood doesn't coagulate. You know, all of these things, there are proper ways that they do. Sometimes as a nurse, the person does it. Sometimes depending on where you are, the doctor is doing all of this. But eventually it gets to a lab tech, and typically lab techs are responsible. Let's say you have gotten the sample and the sample they have grown a bacterial unknown for you, and then that's the unknown you're getting. When we use the diagnostic techniques, most of the time, or basically in general, one of the first things that they do is they grow them and they look at their phenotypic characteristics, right? These are physical characteristics of the bacteria based on the genome that they have, so we can actually look at their phenotypes. One of the easiest ways is you grow it, you isolate it, you look at the bacteria. Is it white in color? Is it kind of ivory in color? Is it glossy? Is it not glossy? The borders that it creates, is it the nice round border? The single colony, is it concave or convex? So all of these things are different. Is it producing a pigment? Does it have a special characteristics? Like for example, for the things that we so far looked at, Pseudomonas aeruginosa has this like gray color to it, and it releases the green gray color to the media. Proteus vulgaris, I've been showing it to you guys constantly. It makes the media kind of look like that sick yellow color. So it's just how it grows. It causes the media to look like that. Staphylococcus aureus, our Staphylococcus aureus has a yellow hue to it, a golden hue to it. Our E. coli almost grows and has like a translucent or very shiny color to it. Serratio marcensis was pink, red. So all of these different kind of characteristics. B series growth, very fast, very fluffy. So you should assume if yours is B series, you have abundant of growth, all right? So that is part of physical characteristics, macroscopic. Then we have phenotypic characteristics, microscopic. This is pretty much the first test that anyone would do when they are trying to identify a bacteria. Gram staining, okay? So you gram stain it, you find out whether it's gram positive, gram negative. Is it a rod, bacilli, or is it a cocci? Like I said, in this lab, we never look at any spirillum. So it's either cocci or rod. It's gram positive or gram negative. We did not work with acid fast, so you would not expect to see acid fast. So gram positive, gram negative. Now, is it bacilli, individual bacilli or rod, or is it in a chain, streptobacilli? If it's cocci, is it individual cocci? Is it staphylococci? Is it streptococci? All of those are characteristics for you guys. So you will look at it that way. Other things that in other labs, which we're not going to do, but they can look at the, not only cell wall, they can look and see whether it has a flagella, does it have a capsule. For them, those things can be used also. But in this lab, we're sticking just to gram stain and morphology. Then the other phenotypic characteristics, remember, it's the enzymes. What genes do they have for what enzymes? So those were the biochemical assays that we did. So for our unknowns, you're going to do biochemical assays. And all of these biochemical assays that we've done so far, you have access to all of them. And that should allow you to distinguish. I'll come back to unknowns, but again, immune testing is possible too. This is what we're going to do with ELISA, and when we do it, I'll go through it and explain it to you. In the other, you're not going to do it. In other labs, we can do the, this is the immune testing. Genotypic methods. We did that already. PCR was a genotypic. So for many of them now, they actually, for a fast response, especially for viruses where you don't have the physical, the phenotypic characteristic, the microscopic characteristics, this is what they're doing. They're doing the genotypic methods. Like the PCR we did for COVID. Using PCR is a fantastic way of, by using special primers, you can actually figure it out. They can actually sequences. They can create probes. So there are all these different ways that you can do it. All right. So what you're doing with your unknown is almost like what they should be doing with identifying or diagnosis. All right. Sticking to your unknowns. So this is the PowerPoint that you have. Like I said, you're going to get your unknown in a slant. It will have a number. Okay. So on Wednesday, every person is going to get their unknown. It will have a number. What you have to do is make sure you put down that number for yourself and then start looking at the macroscopic morphology. Take a look at it. Jot some notes. Okay, does it grow fluffy? Does it have this like pigmentation that I see in the slant? Things like that, all right? And then we're going to follow it by gram staining. Now, when you do gram staining, I gave everyone this chart. It's actually the updated chart. The one that was there, I changed it today, is for spring of 23. Based on the results that we've been getting, I changed them. So, this is basically the chart that you're getting. I could highlight it better as an Excel, so I used an Excel. You have a group of gram negatives here. All right, so gram negative rods, E. coli, Enterobacter aerogenes, K. pneumoniae, Salmonella typhimurium, Proteus vulgaris, Pseudomonas aeruginosa, A. faecalis. You've worked with all of them at some point in the lab. So when you do the gram staining, when you find that it's raw negative, this is where you're sticking to with your unknown. It should be one of these, all right? If you get a gram positive, so these are the gram positives. All right. So when you get gram positive cocci, so you have, for gram positive, you have cocci, and you also have gram positive rods. Okay. Pretty much you work with most of them, and the Corynebacterium xerosis is the only one you haven't worked with. Okay. So first off, if you look at it under the microscope, and it's cocci, you have a mix of gram positive and gram negative. What should it be? Should it be gram positive or gram negative?
Gram negative.
No, positive. You will not have any cocci that is gram negative. So if you see a cocci, it's gram positive. Just go with that, all right? So it's not going to be different. It's going to be the same. Whereas it's rod, then it could be negative or positive. If you're worried about your gram stain and you're not sure about it, what other test can you do to tell you whether it's gram positive or gram negative? Snot test, exactly. So you do a rod and it looks weird. Half of it is pink, half of it is purple. Do a snot test. That should tell you for sure, is it positive or negative, all right? Then we're going to, that's the thing you're going to do on Wednesday. So after you look at it, then you're going to do your gram staining. So make sure you read up on gram staining. Slide making a smear, review that lab. Review the lab with regards to gram staining, snot testing, so that you have it. Everybody's going through it. You can actually ask each other, ask me for help, but I'm expecting you to know it. There's all of your 19 of you doing the same test. So it's going to be hard for me to go and stand with someone and keep doing it. But I will be going around. So remind yourself what the procedures are, review it, be ready to do those really fast. So we will do that. We have the microscopes. Then based on what you're getting on Wednesday, whichever group you are, then that's it, right? When you get a gram negative rod, or let's say even if you get gram positive staphylococci. Enterococcus faecalis is actually a, what do we call it? It's a streptococci. So let's say you get staphylococcus. Now you wouldn't know, is it aureus? Is it epidermidis? Which one it is? So you will switch to all the biochemical tests that we have. There is 2 tests that I actually reviewed for you guys, or I demoed for you guys on last week on Wednesday, that the results were immediate and fast. Do you guys remember what those were? Oxidase and catalase, thank you. So those were pretty fast and immediate. So can you do those on the same day on Wednesday? Yes, I will have those there for you too. Now, but should you do them? Depends. So look at the gram-negative rods. So looking at catalase. Is it a good test for a person who has a gram-negative rod to do catalase? Does it give you any information? No. So don't even go near it. Something happens and that catalase is wrong, you're not getting the right result, all it's going to do is drive you nuts. By the way, I pretty much write down next to the numbers based on what Kim tells me. whether it's gram-positive rod, gram-negative rod, gram-positive cocci, and so on and so forth. So that I know. The rest I don't know. I give them out blind, all right? I will, which my colleagues tell me I'm insane, I will confirm for you whether it's your gram staining result is correct. So once you're completely sure, you come to me on Wednesday and you say, Sina, my unknown number is unknown number 5. And I believe it's gram-positive cocci. You can actually ask me to come and look at your slide. And then I'll look at my list and I say you're correct. You are a gram-positive cocci. That I will confirm. After that, I don't know. I can't tell you whether your catalase result is correct, your citrase result is correct or not. I don't know. I'm going with it blind because I don't want to push people into the areas. Okay? So, in this case, the catalase is basically is not a good test for you guys to do. So don't do it. When it's not a good test, don't do it. All right? Now, but when you look at it, oxidase is pretty fast too. Look at these two. The oxidase for 2 of them is positive. So right away, if you do oxidase and your oxidase comes out positive, you know you're one of these 2. Is your oxidase negative? You're one of the ones above. So that's a good test to do. When it comes to the others, like the gram-positive, catalase is pretty much the only one that you can do for the gram-positive cocci. But if you're even staphylococcus, don't even worry about it because catalase for both of them is positive. The only one is this one. If you're not sure about morphology, maybe that would help you. For the gram-positive rods, neither of them are good. Okay? Catalase and oxidase for both of them is the same. Then what I want you guys to do at the end of Wednesday. So Wednesday is going to be actually a long lab. I want each person to sit there, look at their chart, and basically highlight all the possible ones. So let's say you are among these 2. Okay? You are among these 2. This. Okay. You are among these 2. So you want to see which one is the best one. And I love that we have this page thing there. So you look at it and you say, okay, lactose, sucrose, none of these help me. There is not much of a difference there, not much of a difference there, nothing there. So the first big difference is going to be citrase. That's a good one. Then gelatinase, that's another one. Milk agar, that's another good one. So you choose those. By the way, you can actually use other things. EMB, McConkey, mannitol, blood, all of those you can use. Blood can be used for any bacteria. All you have to do is just research out there what is blood. I have not put the results there. So blood for McConkey, if you're having a suspect pathogen, which one it is. So those can be also put there. So any person that is suspecting any of these has to come to me by the end of that lab or whenever they're ready and say, sign up. I'm ordering these media from you, okay? So you are going media shopping with me. You say, for me to know what this bacteria is, I'm going to need to do citrase activity, I'm going to need to do gelatinase activity and casease activity. But search and find the name of the media. You can't come to me and say, I'm going to do citrase activity. I'm like, great, what's the media you need for that? So you order a Simmons, okay? Then you order gelatinase, gelatin for me. Caseous, milk agar, okay? Anything like blood or anything like that, if you want, again, you can order. So you say, I need one of these. I'm gonna tabulate what each person needs, and I'll go through it. For some of these, it's hard to find more than, like for that, you can only have 3 tests, really, that distinguishes them. For the staphylococci, really these two, so a little bit here, MRBP, these two are almost the same, so MRBP is a good test. What else is good for Staphylococcus aureus and epidermidis? What can I order that it's not there? And it will give me a result after incubation. Crazy result.
Mannitol salt.
Mannitol salt, yes. So I usually say I want 3 or more tests, but for some of them, I totally understand. You cannot ask for more. For between those two, really, you can only ask for 2 tests. You can add blood to it too for the 3rd one, because remember, aureus did a beta, whereas epidermidis did an alpha for us. So you can again do that. So you come and ask for those, and I will have those ready for you next Monday. Then on Monday, all you do, it's a fast lab, you're gonna come and inoculate. Hopefully, on Wednesday, you get all of your good results. If not, we have more media. You're like, okay, these 2 are weird, I wanna try it again. Okay, we'll try it again. I'll get you more media. The other thing I wanna tell you guys when it comes to these is, if it is, for example, I always say, if it quacks like a duck, waddles like a duck, it is a duck. Now, you're used to seeing that brownish color, it's a purple duck, it's a purple duck, okay? Just because it's a purple doesn't mean it's no longer a duck. Somebody's stained that poor thing purple. If all of the tests, like for gram-negative rods, you can actually ask for a lot of tests depending on which areas. These that are up here, you have a lot of differences. Remember, we have TSI, and then in addition to TSI, we have the sucrose one too. So you can actually check that and see the differences in sucrose. How can you check lactose fermentation, whether they're a lactose fermenter? What media would you use? EMB or McConkie. Good job. If you think you're having E. coli, what would put your mind at rest?
The one that turns metallic green.
EMB.
Yeah, EMB.
So if you're one of those, why not do EMB too? But if you're doing everything, and everything points that this is E. coli, except, for example, the MR came out negative. Everything else is saying it's E. coli. It's metallic green and your MR is negative. Should you care? No. Somehow my MR came out weird. You can actually use that in your discussion and say, everything pointed, this one, possibilities, I didn't inoculate enough, maybe, somehow something happened. By the way, when you're doing this, grab a loop for yourself and keep that. And the first time you're using it, you inoculate the heck out of that thing. I mean, not inoculate, sterilize it, thank you. Incinerate it to make sure there is nothing on it. I will give you guys a plate. So these are the ones, she started growing them today. So you will get a fresh one on Wednesday. We will give you TSI or TSA plates too. What you're gonna do on that plate, try to do streetway too, to make sure that you have pure isolate. You're going to grow that for your Wednesday, no, you're gonna get it for Wednesday. So we're gonna transfer it to that, and then as soon as that grows for 24 hours, I will take those out and put it in the refrigerator. So if you come back on Monday, you will have your plate with tons of bacteria on it, and you will have single isolate colonies that you could go to, or if you see basically everything looks the same, all the other areas, but I will keep it in the refrigerator for you, and I will give that so it stays fresh, so your results would look good for your biochemical assays. Your biochemical assays, again, on that day, Monday, when you're starting, make sure your loop is nice and clean, all right? Then when you're inoculating, heavily inoculate. Take a big goop of it and add it to MRVP. Take a big piece of that and put it on EMB, okay? You want to make sure that you transfer good things, a good amount there. All right, let's see. Okay. Now, as far as reports, I will bring copies of the reports for you on, I didn't bring it today. I will bring copies of how I want it done, reports for unknown, for you guys on Wednesday, and I will keep having them every day so you can take a look at them. You can't take them home because these are from previous students. I don't want to put them online because, again, it's their work, so but you can take a look at it. The reporting is going to be basically done like I have given you this as a research project, as a lab tech, and you're doing it. And in a research project as a lab tech, notes are taken every day, and pretty much like a conclusion is made every day, and what you do the next day is based on your conclusion of the previous day. That's how I want this to be done. So on the first day, you would write on it, this is unknown lab, your name, the date, first day. Your introduction part of it is basically a purpose. You put it on the first day. What I am doing is I have received unknown number 5, and I will be going through series of biochemical assays and phenotypical assays in order to identify my bacterial unknown as one of the possibilities or whatever that is given to me by the instructor. Purpose of biochemical assays is to differentiate and eventually identify a single bacteria. Great, purpose. Then, write down unknown number 5, physical characteristic. Received a slant. The slant has a lot of growth. The slant's coloring is has a golden hue. Pseudomonas aeruginosa, some pigmentation has been there, things like that. Methods and material. Today, the experiments that are going to be done, in addition to just observing the bacterial growth, is gram staining, possible snot, and oxidase or catalase. So you might not do that. So start with methods, gram stain. Create a bacterial smeal. Very short, tell me what gram staining is. In your other method, you also underneath say possible snot test. Again, tell me what snot test is. Again, you can do these later on. If you do oxidase, add what oxidase is. If you don't do snot, don't add it there in your report. Just do gram stain, and I did oxidase. My gram stain came out beautiful, all right? So you can kind of fudge it later on. Move it in your methods. So methods is a very fast and dirty of this is what I'm gonna do. Results for your Wednesday is, create a table for me. Bacteria, gram staining. Bacteria was gram positive, purple, streptococci, okay? If you did snot test, not, and then like oxidase positive, turned blue, this fast. That's your results, all right? Leave it, now you go to discussion. In your discussion, you basically write down, based on what I have seen, and based on the table of unknown characteristics that I have, My possible bacterial unknown. So we said gram-positive staphylococci. My possible bacteria, since it's a gram-positive cocci, is one of these three: Staphylococcus aureus, Staphylococcus epidermidis, Enterococcus faecalis. Due to the fact that my bacteria was a Staphylococcus, and let's say you did, what was it, catalase, and it came out as catalase negative, therefore it tells me that my bacteria cannot be Enterococcus faecalis. Then the possibilities are Staphylococcus aureus and Staphylococcus epidermidis. Based on the possible biochemical assays, my next step is I am going to conduct such and such tests, okay? Mannitol, this, that. That's your day 1 conclusion. You come back Monday, your day 2. Your day 2, again, you don't have to write purpose, you start with methods and materials. So you're doing mannitol, tell me how you set up mannitol agar. You're doing gelatinase, tell me how you set up gelatinase. What agar you're using, what is happening, so on and so forth. Your conclusion on that day is not much. I inoculated, I have to incubate everything for 24 to whatever hours. I will look at my results on the following day. Day 3, which will be your next Monday. Again, you come, you get your results, you just start with results. You don't have any methods and material at that point, you start with your results. Let's say it's indole. So in your results, say after COVAX. So for results at the top, say indole results was read after COVAX was added. Gelatinase was read after it was put in the refrigerator. So make sure that you put that, then create another table for me. Then in your conclusion, if everything went well, on that day you can actually figure out what you have. So based on the results from day 1, my possibilities were Staphylococcus aureus, Staphylococcus epidermidis. The mannitol, because of the results of like gelatinase and mannitol and this, it indicated that my bacteria is Staphylococcus aureus because of blah, 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 blah. Great. You're done with that. 2 more things that are left. You need a paragraph about Staphylococcus aureus, for example. What is Staphylococcus aureus? Google, use your book, find a little tiny paragraph from introduction. Staphylococcus aureus typically is found in these places. In humans, it can be cause of such and such diseases. Find all the diseases that you can have Staphylococcus cause it. A little bit of intro. What are the chances of people getting this? Where is it found? So things like that. Just a little paragraph to tell me this is a horrible thing to have. This is, normal flora. Staphylococcus epidermidis, if it's a normal flora of humans. It's typically found on our skin. Under normal conditions, it shouldn't cause any disease. Otherwise, it could be opportunistic and cause blah, 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 blah. Okay? So see how it progresses and it goes. If you need to go to another day, do additional stuff, fine, we'll go to day 4. Because of the results that I got, I was still questioning between these 2, so now I'm going through it. So at every step, like this was just a few, but for gram-negative rods that are oxidase negative, all of those, 5, 6 that you have to, in your conclusion, step by step, keep eliminating and tell me eventually because it's this. One last thing you do there at the end is what we call a dichotomous key. So taxonomy, remember, is a science of classification. And basically most of the time we do it based on phenotypic characteristics. Carlos Linnaeus came up with that. Now we actually can create dichotomous keys. A dichotomous key, taxonomic keys, dichotomous keys. Dichotomous keys, sometimes if you've gone to like Redwood Forest and you've gone to the forestry station, there's like books that they actually give, like they grab kids, and one of the rangers takes a bunch of the kids with these little booklets and says, okay, let's identify this tree, let's identify this bush. Dichotomous means two-part. So you can actually identify a species based on answering these two-part questions. And the two-part questions are, it is this, no, go here, it is it, yes, go there. So a question has a two-part. Let me give you an example. So I want you guys to create a dichotomous scheme. So for example, I have up there, feathers, yes, you go to that other question, no, you go to the other one. If they have feathers, does it swim? Yes, it's a duck, no it doesn't, it's a hen. All right? For example, again, dichotomous key is two parts. You can create it for anything. You have an alien friend that is visiting the planet and you want to make them a fruit salad. So you don't have time to go grocery shopping. You tell your alien friend, go grocery shopping, get me a banana, a red apple, red grapes, and a strawberry. Okay. So your alien friend says, all right, what is a banana? What is a strawberry? A dichotomous key can be made for these. So you look at their physical characteristics, and based on how each individual organizes things and differentiate things, you can have many dichotomous keys. There are so many dichotomous keys for trees. People who bird watch, each of them have a favorite dichotomous key book for their bird watching. Like they have looked at all the books and they're like, this one, I don't like how they organize it. I always get lost. But this author, I like their dichotomous keys. It's separated. Looking at these, you can separate them based on shape, based on color. Depends on who goes and decides what question is first. Like, I can say, is it yellow? Yes. Or is it not yellow? So for yes, I say banana. I can use shape. Is it cylindrical, oblong, or is it round? Then again, if it's cylindrical, I can say it's banana. So let's use color. You can say yellow. So you can write it this way, or you can create questions. So how I want you guys to write it is this. So we start by question 1, yellow. Yes, no. It's yes, therefore it's a banana. I'm not going to go even differentiate it any further. Nothing else is yellow there. No? Then let me find some other difference there. I don't know. Some people can say it has the seeds on the outside. It's heart shape rather than round. It has a pit. It has multiple. It's a cluster of round fruits. However way you want to make this. So, so I'm going to say, is it cluster? So this is the other question. You keep the yeses and nos in the same side. Yes, it is a cluster, so therefore it is grapes. No, it isn't. Now I have, so I've gotten rid of this, it's these two. Red apple and strawberry. Seeds on the outside has a pit. I can say large or small. Let's say, which are, let's use this. Seeds on the surface. Yes, no. It's strawberry, it's an apple. I'm done with my dichotomous key. For you guys, when you're going through this, you basically will start with the grams thing. So you say, is it, so you can say gram, positive, yes or no, or you can actually start with this one with going positive, negative. It's up to you. Then let's say it's gram negative. From the gram negative, you do, the next thing you did was oxidase. Or is it rod or cockeye? Again, you can start it like that. You can start by just saying it's a gram negative rod. That is an acceptable start for you too. So that's my first, that's my initiation, gram positive, gram negative rod. It's one of those. The next test that I did was oxidase. Oxidase, did it have oxidase? Yes, no. If it is yes, it could be Pseudomonas aeruginosa, I'm not going to write the whole thing, or Alcogenes faecalis, but you guys should be writing it. If it is no, it's E. coli, it's Enterobacter aerogenes, and so on and so forth, okay? Let's say we are no, so it's one of the 6 or whatever that is. Then you do another test. So the next test underneath the no is citrase. So citrase activity. Again, keep the yeses in one area, yes, no. All the ones that are citrase positive, so citrase activity, Enterobacter aerogenes, K. pneumoniae, Salmonella typhimurium. All right, so that's because we are just among those. 1, 2, 3, 4, 5 of them. And then on here, we're going to have E. coli. P. vulgaris is positive, negative. So it could be one of those. This wasn't a good choice to have right here. But let's say it was positive. So if it is negative, you're done. You can actually add more tests here next to it. Say, in addition, EMB was metallic green or whatever. If you're not here, between these 3, let's say endol production. So endol. Yes, no. The endol production is, the only one that is positive is P. vulgaris. The others are negative. If it's positive, you're done with P. vulgaris. If it's negative, you kind of have to separate those 3. Does everybody understand what we're going through? When you play with it at home, might sound crazy. Doesn't have to mean, you can actually organize the order of the tests any way you want to make your dichotomous key pretty. If the same things are yes for it, like it's indole positive, citrase positive, and all of that, it's just individual, put it in one area. That's okay. So again, let's say you did more than these tests. You don't have to put all of them on the dichotomous key. You can choose, all right, out of the 5 tests that I did, in order to make a pretty dichotomous key, I can use 3 of them and it would be very easy and simple. Just use that. All right?
It's a process of elimination.
Yes.
And you can see it. So
It's basically how you use it in your conclusion, the last conclusion, how you worded it. You just need to create it that way so I can go through it and say, got it, you did good. All right? And again, when you go through this, it doesn't mean that you can't come and ask me and say, does this make sense to you? Do you think I did this right? You can ask questions. All right?
Would you like us to add pictures in our results for our everyday reports?
You don't have to.
Okay.
Some people like it. It's again, those are like your little preferences.
Okay.
Some people right away when they have unknown number 5, they take a picture of the slant or their street plate. But tables are more than enough for me. How you want to write it, you'll see from the reports that I have. Somebody starts right that way. They didn't do any head cover or anything like that. In my unknown report guy, I say do it APA, but APA is technically for the citation part, what you use to get me that information for the last paragraph talking about your unknown, what it is, just give me a citation for that. And that can be APA format. But style of it, how you want to do it is different. If you want, I look at this at the same time that I do your second report check. So for those of you guys that keep a lab notebook, just make sure you leave a few pages for your lab 13 report, then do your unknown at the end. And lab 13, probably you would need like 2 pages, nothing more than that. And then start your unknown and do the same format, same results. If you like to write a pretty note, you can say notes for unknowns and then start your report at the end when you have it. So you can put it in your lab notebook. If you want to type it, type it. Okay. All right, guys. So that's our unknowns. I'm gonna give you guys, if you want a few minutes, you want to go get coffee, whatever. If not, how many of you guys want to go off campus get coffee? Nobody. All right, so